All right. Welcome, everyone, to the Cornelia Klopp Auditorium. I need to tell you two things before we start. First is that this event is being recorded, and the second is please turn off your cell phones if you haven't done so already. So I would like to welcome Paul Turner, who will be presenting this week's Friday evening lecture. And it's my pleasure to have the opportunity to introduce Paul. My name is George O'Toole. I'm the Elmer Pfefferkorn Professor of Microbiology and Immunology at the Geisel School of Medicine at Dartmouth. I'm also co-director with Rachel Whitaker of the Microbial Diversity course here at the MBL. If I was giving this introduction 2 years ago, I might have started with the line, "I bet you don't think much about viruses." Well, that is not at all true, obviously. But I think I'm safe in saying that I bet you don't think much about viruses of bacteria. Paul has worked on viruses that infect creatures, from animals to bacteria, and how these viruses impact the ecology and evolution of the creatures that they infect. I think if you asked Paul 10 years ago whether or not he would anticipate giving the talk that he's giving tonight, I think he'd probably say no. A talk that focuses on viruses to cure infections, which is a nice change of pace. Dr. Turner's work illustrates perfectly, I think, why the American people and people around the world have made a commitment to supporting basic research. The studies that Dr. Turner performed early in his career, understanding basic and fundamental processes by which viruses infect their hosts, did not necessarily have obvious connections to human health. But what he learned in these early days set the stage for the impactful work that he's going to talk about tonight. And there are plenty of examples of how basic science leads the way to improve human health. From the basic studies at my institution at Dartmouth that directly led to the coronavirus vaccine that most of you, I hope, have flowing through your veins. And of the science going on every day at the MBL, advancing our understanding of neurobiology, regeneration, climate change, marine science, and of course, the microbial world. The commitment to basic science is an important one, and so thanks to all of you who have supported that work. Dr. Turner's career is a highly decorated one, and I'd like to highlight just a few of the awards that I think reflect the Paul Turner that I've come to know. He was invited, or has been invited, to numerous named lectureships, including the Roger Milkman Endowed Lecture at Microbial Diversity this year at the MBL. He's president-elect of the International Society for Evolution, Medicine, and Public Health. Evolution, medicine, and public health. Think about that, and the diverse topics that spans. I think that's really impressive. He has been elected to the American Academy of Arts and Sciences and the American Academy of Microbiology, and he's the Rachel Carson Professor of Ecology and Evolutionary Biology at Yale. He's been named the E. Just Endowed Research Fellowship and William Townsend Porter Award here at the MBL, and was named as one of Cell Press's 1,000 Inspiring Black Scientists in America. He's published more than 125 papers in his field, and his most recent award is his election to the National Academy of Sciences. And being a member of the National Academy reflects an outstanding career of scientific achievement. But I also think Paul sees as one of his major contributions his commitment to mentoring from undergraduates to newly minted assistant professors. I'd like to finish by saying that there are scientists that do transformational and impactful work, and there are scientists that are great speakers and teachers. But there are a few that excel at both, and Paul is one of those people. So I think you're really in for a treat tonight, so enjoy.
Well, that was a fantastic introduction, and I hope I can live up to it. I really appreciate that, George. Thank you. I'll also try to get this. Are you good for me to start screen sharing? Okay, actually did that without a hitch. Thank you all for being here. I wanted to begin with this thank you slide. I have to admit, it's a little surreal for me to be on this stage. For many years, actually 20 years in a row, I've been coming to Woods Hole in various capacities. A lot of it has been to participate in courses, the Molecular Evolution Workshop initially, currently the Microbial Diversity course, and through various research stints in collaboration with the folks in the Bay Paul Center, pictured here, especially Mitch and Hillary, This is just a place that has inspired me in my career in so many invaluable ways that I have a hard time expressing that. I was literally this morning sitting next to the Rachel Carson statue, and it's lovely how it says how she's gazing out at the water, inspired by the location that she's in to do her work as an environmental scientist. And I've just done that for 20 years, and I can't calculate how valuable that has been to my career. So I'm very proud to be the Rachel Carson Professor. When I learned of that as an endowed professorship, not only to get that at my university, but to be named after such an amazing person for my professorship, that's quite amazing to me. So thank you all for being here, and hopefully in the next 40 minutes or so I can explain to you this one project that we're doing that seems to be now boundless in its possibilities. So this is my first slide, and the idea here is for you to understand with these three pictures essentially the punchline that I'm going to say today, and that it is very possible to go out in natural environments and to find viruses that are specific to bacteria, and they're called phages. I'll talk about them tonight. And this is a member of my research team, Ben Chan, doing exactly that. He happens to be in Mexico in this picture. In the middle is a picture of one of the viruses that we've found in nature, one of these phages, and it has some characteristics, properties that I'll describe that made it possible for this last picture to occur. This is a young woman. I'll tell you more about her case later on, but she's in a hospital room in Texas undergoing pulmonary decline, ultimately probably failure. So her lungs are giving out on her. Why? Because she has an antibiotic-resistant bacterial infection, and technically biomedicine, there's just no hope for treating it because it's a pan-drug-resistant infection in her case. No known antibiotics are able to kill her bacteria. So the same person pictured on the left is showing her a vial of phages that over the course of several days, she's going to inhale them with the hope that those would kill the bacteria instead when antibiotics cannot do that. So this is the kind of work that we've been trying to do lately. It has a particular punchline to it having to do with evolutionary biology, and that is my training. So I'll explain that. But essentially, this is where I want to take you today. This is the problem, one of the many biomedical problems that is looming in the background. It's an ongoing one. We're spending so much time and attention, deservedly so, on the pandemic and pushing back on it. Sadly, it's a reminder that this is something that is not going to go away. It's only going to get worse. And it goes like this. I'm assuming this audience, maybe our listeners as well, very literate group, you understand that we have a big problem in biomedicine with the increasing proportion of bacteria, both pathogenic and not pathogenic, that show resistance to commonly used antibiotics. And it's not going on in one country or one region, it's around the world. This kind of a projection map out a couple of decades from now is very sobering because it's saying that this is going to be a leading cause of death in the human population unless something changes. So in the most populated parts of the world, we're going to get the most annual deaths, but essentially it's a global problem. Many reasons I could talk about for this problem. One could ask, well, how come the pharmaceutical industry is not doing more? They have tried, and a lot of amazing discoveries through time have happened through big pharma to develop, discover new antibiotics, and that takes millions of dollars. And the problem is if they do it now, literally, these are seen as last resort drugs, and they're placed on the shelf, and they actually don't go into people unless there's a desperate need for that to happen. So if you add all that up, there's no money in that. By the time you approve it, there's so much money spent, you don't really get that money back because the drugs sit on a shelf. So we're left to the academics and other people, academia, to try and create these drugs. The point is it's very hard to find new antibiotics, and we would like to look for alternatives. These are a few more details of the woman who I showed you earlier. She's allowing us to use her name, Paige Rogers. At the time we came to know of her problem, she exemplifies what I'm talking about. So we have this antibiotic-resistant bacteria threatening lives, and in her case, I'm giving you the details. She's a young woman, but she has a genetic disease, cystic fibrosis. I'll talk about that later, but it makes her more vulnerable because of her lung architecture to certain bacteria coming in, causing chronic infections. And if increasingly those are antibiotic resistant, that's a collision course to a bad outcome. It can lead to pulmonary failure, lung failure, or death. So she had been battling this for literally her entire life, 20 years. She's only 22 years old at the time that we knew of her. And this gives the details of how she spent nearly a month in the hospital and was finally discharged and went home, but it wasn't because anything was done to help her. Her physicians had tried, and there just were no antibiotics available to help her problem. So this is sadly, you hear this again and again and again with increasing regularity. What are you going to do about it? So this is literally an old adage that I'm not a historian. I don't really know where this came from. The best that I could do was to figure out some 4th century BC, circa this proverb was stated. The enemy of my enemy is my friend. And what I'm going to describe now is if you have a persistent problem like drug-resistant bacteria, can you turn to their enemies to help solve that problem? And that's where these bacteriophages come into play. So this cartoon here is showing a lytic phage replication cycle. The only details you need to understand is that this is a virus called the bacteriophage, literally translates to eater of bacteria, and it's interacting specifically with some cell type of bacteria, overtaking the molecular machinery to turn it into a virus production factory. And it is a lytic bacterial phage, so that means it bursts the cell open to liberate these particles that can then go on and cause the same kind of an interaction with the bacterial target. It's very attractive in terms of killing bacteria. So long before antibiotics were even discovered, some of the early co-discoverers of phages were testing them in animals and in people to cure infections. Felix Durrell is a co-discoverer of phages, early 1900s. He was doing that work. He happened to have only one faculty appointment in his career, and it was at my institution at Yale. So in a sense, we're trying to carry on some of his work. He was instrumental in the early days of an institute that still operates in the country of Georgia called the Eliava Institute. So he helped George Eliava found that institute. Ultimately, George Eliava was killed by Stalin because he was seen as an enemy of the people. That's a different talk for a different day. But you should know that this work is ongoing in places like Tbilisi, Georgia. It never stopped. If you want to learn more about Felix Durrell, this is a very nice book written by one of my emeritus colleagues, Bill Summers. So what are the benefits of phage therapy? There's some pretty obvious ones. Specificity to target is a good one for drug development. If you know your drug is going to work on some target and not another one, that's attractive. Self-amplification is very hard to come by in drugs. And this is a case of a drug that's making copies of itself. So you could use minimal dosing, for example, to achieve a good outcome. But with those benefits, there are also some pretty obvious costs. If it's too specific, it gives you too limited spectrum of killing ability, and that's not very efficient. The delivery to the infection site, if you're treating the lungs, an infection in the gastrointestinal system, et cetera, there are different ways of trying to reach it, and it may be easy or hard, depending. An ongoing area of research is whether your immune system catches on to this. So it's certainly the case, I'm very confident in saying that today you ingested phages. You drank them in water, you ate them with your food, you breathe them in as you're sitting in this auditorium. Those are here present with me because they're attached to dust particles in the air. And you don't really mount much of an immune response to them, and that's not that surprising. It's sort of like the human species and many others evolved in a world that's kind of full of phages. But it's different if I'm giving you 10 to the 10 of them, 10 to the 10 of them every 3 hours for 10 days in a row. I would think your immune system may catch on to that being a foreign invader. But the last cost of phage therapy is that I'm always predicting it's going to fail. As an evolutionary biologist, I appreciate that bacteria are very good at evolving. They've evolved antibiotic resistance quite well, thank you very much. And they should also be capable of avoiding this kind of therapy because they evolve to withstand it. And there are lots of ways for me to show this. This happens to be a couple of cartoons from a recent review article by someone else, but here on the left, these are what people tend to call innate mechanisms of resistance in bacteria. So things that might at the cell surface by proteins that are changing that would ordinarily be receptors for the phage to attach to, they evolve away from that morphology, and therefore the phage can't bind to them anymore. If the phage genetic material gets in the cell, it will be cut up by restriction enzymes, etc. On the right, This is a more elaborate thing that you might have heard of, CRISPR-Cas systems, where this is something more recently discovered, and essentially it's a way for a bacterial population to build up a recognition of phages that ordinarily would attack it, and they are immediately capable of deploying defenses against them. So all this stuff stands in the way of phage therapy. And the question is, could you use phage therapy in earnest to help solve the antibiotic resistance problem, or are you always going to run up against this? So where we try to take our evolution thinking into this problem, and sometimes people call that evolutionary medicine, is how do you take evolution thinking to human disease or disease in any system and try and think about it in a different way and come up with maybe better therapeutic options. So something that you see quite regularly if you study evolutionary biology is that organisms don't evolve traits that make them universally good at everything. They tend to evolve traits that they need to function in their environments, but it might make them less good at doing something else that they might have done in the past or compared to other related species. So there's lots of ways for me to talk about this, but I feel like with human audiences, I should talk about human biology. What do we do in the animal world that is better physically than other organisms? We throw objects with speed and accuracy. And the best of us do it professionally, as softball players and baseball players. Chimps can't do that. Neither can we, for these close relatives in comparison, hang from a branch with a single arm from an extended period of time like this young chimp is doing. So we just have different musculature that allows us to function in our environments in different ways. But if you just look within human biology, there's many quirks of being a human that make us more prone to problems. And being the only bipedal great ape is problematic, because we're walking around all the time, and we also have a tendency to suffer lower back pain and neck pain because of it. And I don't think chimps and gorillas suffer through that. At least they don't complain about it. So this is something unique to humans, but it's a reminder that humans are by no means some pinnacle of evolution. We struggle with our own traits in a way working against us to do other things well. So what does this have to do with phage therapy? So essentially, we took that idea to phage therapy to consider if you choose the right phage, you can probably paint the bacteria into a corner where they evolve phage resistance, but they suffer a trade-off alongside. So I'll walk you through a couple of examples of this quickly. Imagine this is a bacterial cell, and these orange and red things, these are efflux pumps. They're complexes of proteins that if the antibiotics make it into this cell, these are actively pumping them out. And this can make some bacteria multi-drug or even pan-drug resistant to antibiotics. They're very efficient at doing this across different drug classes. If you found a phage, this is what we thought. If you found some phage that evolved on this planet that is naturally using the outermost protein of efflux pumps as its receptor, then this bacterial population has a big problem. They're basically going to die of phage attack, and evolution will allow them to be selected to avoid that problem. But meanwhile, they could get rid of efflux pumps as a result, or downregulate them, or change them somehow so that they shift more towards the bottom of this screen, and instead of being antibiotic resistant, they become sensitive. So it's like a two-edged sword. You kill the bacteria in the traditional sense of phage therapy. Meanwhile, you're selecting for the leftover bacteria that evolve resistance to be more vulnerable to biomedical therapies. So I'll be faster in explaining that. Imagine this purple surrounding this cell is a capsule that shields it from detection of cells in your immune system. If you have a phage that is specific to it, again, it's going to kill the bacteria. It'll select for mutants that avoid that problem. They'll get rid of the capsule, and they'll become less virulent because they're easily seen by the immune system and targeted. So we had this idea, and we said, okay, we could engineer this, or we could simply go look in nature to see which of the phages that are so abundant on this planet, they're the most biodiverse thing on the planet. Probably some phages have exploited these very many receptor sites around bacterial cells that we care about as pathogens, and they also function as virulence factors and antibiotic resistance mechanisms in these bacteria, and we're just going to go find phages that exploit those. Okay? So keep that in mind, and I'll try and convince you that it's actually possible. So I'm going to walk you through what we call our discovery pipeline. How do we go to nature all the way to characterization to find something interesting and then use it in people? all right? So how does this happen? The first thing is, I guarantee you phages are exceedingly easy to find. So you can go outside, literally steps away from this building. You can probably find them in this building pretty easily. You could go to very pristine environments like this lake in Connecticut. This was one of the first places that we found a very promising phage. You could go to human-altered environments in a very radically human-altered way, like this coastline of Haiti, or this river in Democratic Republic of Congo. And the classic place to look for phages is where you find bacteria that are associated with people. And that would be in like a wastewater treatment plant, sewage treatment plant, or on an agricultural farm where we use antibiotics in earnest. Therefore, there's a lot of antibiotic-resistant bacteria, and maybe we can find phages that would target them there. So I would say phage hunting is pretty straightforward. Once you have these phages, the next thing you want to do is, in my opinion, figure out genetically what they are. They might harbor genes that are not so good for therapy. They could carry some toxin genes or something that you deliver to the patient, and that's not going to do the patient any good. So what we always do is, in this example, we've got over 100 phages specific to Pseudomonas aeruginosa, which is an opportunistic human pathogen, and we're separating these out into known virus families based on their genetic relatedness to one another or not, and their morphology, et cetera. So all this gives us information that in the classic days of phage therapy, they had hardly any understanding of really what a gene was, and this just was not happening. This was one of the things that worked against phage therapy, so that when Fleming accidentally discovers antibiotics, the Western world just bought into that. That's not something messy and biological. That's a chemical entity that you can trust to not change on you. So the point here is that you can do this because genetic sequencing and genome sequencing is increasingly affordable. Now, one of the problems of phage therapy, at least an assumption, is that you're going to find phages easily, but they're probably so specific to some even genotypes of your target bacteria that they're not going to be very useful for therapy. I don't want to go find a phage for you, and a phage for you, and a phage for you every time you come into the doctor's office with a problem. So we've done, and others have done, experiments where you take phages straight from nature and you fully sequence them to know what they are and their relatedness. That's here on the left. And you could challenge them with different strains of bacteria that are taken from human disease, straight from the clinic. And you can ask, if I use this phage against this problem, is it only infecting one or two of these genotypes? And the answer is no. In this one example, all the dark blue in this matrix is when the phage on the left could kill the bacterial isolate on top, that for all I know, it has never ever seen it in the history of the evolution of that phage. So that gives me some confidence that I can go find them, and maybe I can do the genome sequencing on the bacteria to help explain to me why some of these bacteria are susceptible and why some are resistant to these phages that we're challenging them to grow in the presence of. Now, it tells me already that some viruses, including certain phages, are naturally broad in their what's called host range, their ability to cover genotype space in these target bacteria. So that's not an automatic liability to this technology. But if you look now at this one data set in its entirety, there's a lot of information here. I only showed you the very top part of this figure. Along the left here, there's a lot of phages challenged to grow on these bacteria. And again, you're seeing a lot of dark blue. That's good news. But it's hard for me as a researcher diving into the genetics to make sense of which cells are dying when and why. So you have to take more advanced tools to this. And we're using machine learning, artificial intelligence to grapple with matrices of this size and larger to tell us, okay, thank you for that information. Here's a genome sequence of yet another genotype that's not in this analysis. Tell me which of these phages should be able to kill it. That is the future goal of phage therapy, is that you don't want to waste the time anymore matching this up personalized way. You want to be able to have the confidence to move forward faster than that, the way we do with antibiotics. So now we've taken us to the point of we find phages, we genetic sequence them, we find that many of them are broad in killing, but the real thing that I'm interested in is what receptors are they attaching to? And if they're attaching that receptor, I'm gonna be able to predict that those phages are gonna kill the bacteria and exert selection for the bacteria to evolve a trade-off. So how do you do this? The sad news is in virology, it's very painstaking and slow to find out exactly which receptors that viruses use, including in phages. So we tried some high throughput technology ways to do this. This is a paper from 2020 by one of my former graduate students. She's now stayed on for a postdoc with me, Katie Court-Wright. So there's an existing technology where you can take bacteria and expose them to transposons, which will come in as genetic parasites and insert in one and only one location. And they'll disrupt some gene randomly, but they won't disrupt a bunch of genes. They'll only disrupt one at a time. So you create a library. And then if you put it in the presence of some environment that may matter for the function of those genes, like a phage, then what grows out are going to be the bacteria that have genes disrupted that the phages care about, okay? And the output that I'm going to show you in a minute, I'm color coding it here. In this upper left portion in blue, that's going to be the hits that are good hits in this analysis, that we're going to see genes that are important for phage use. Probably we're inferring them correctly as receptors that the phages are using, and we'll be able to at least narrow down what the receptors would be in the system. So this worked very well for a control. A very well-known phage of E. coli called T6 is known for a very long time to use what's called the TSX porin of E. coli. This is a structure on the cell surface. It's binding to it, well known. And this was the most statistically significant hit in this analysis when challenging it to find it. Here on the right is a phage that basically all we knew about it is that it kills E. coli and can grow in the laboratory in the presence of E. coli. And you put it through this analysis, it also turns out to have the same hit. This is not the only hit that you get for E. coli phages. I'm showing you this for a particular reason that I'm going to get back to later. But this is very confidence building. It means that maybe we have a high throughput way of at least inferring what receptors are, and we can narrow down the options of candidate phages that we want to use in people because we know that they're interacting with receptors we care about. So now I'll walk you through a couple of examples, starting with a phage that selects against virulence in a bacterium, and I'll show you an example of a phage that selects against antibiotic resistance. A lot of information here, but on the left, you'll notice this is a similar diagram that I showed you before. This one phage, TIVP-H6, stands for type 4 pilus H6, and we took it straight from nature. We know that it is able to kill Pseudomonas aeruginosa. This analysis says that, yes, we are correct in inferring that the type 4 pilus genes of these bacteria, when you break them, this is what the phages care about, so that is the target that they are attaching to. But there's a cool thing that I will describe briefly, and that is the type 4 pilus made by these bacteria is linked up with a gene network to the production of a nasty virulence factor called piocyanin. And it turns out if you select against the type 4 pilus production, you also select against the ability of these bacteria to make this toxin. Why do you care about that? Because in infection in the human body, piocyanin causes a lot of cellular inflammation and tissue damage that contributes to mortality. So we've got now piocyanin as an important virulence factor with toxic effects. We have a phage that can interact with it, and I can show you in a picture. Piocyanin has this bright green color. When you grow bacteria in the lab and they produce it, you can easily see it's happening. So these are the wild-type pathogen, and if you select for them to overcome phage attack and they're mutants, they're often debilitated in the ability to make type 4 pili and also in piocyanin production, so you grow them up and they look just like normal growth media. They don't make it anymore. So a bonus factor of targeting type 4 pilus in these bacteria is that it contributes to what's called biofilm formation, these very tight structures and chronic infections that is hard for antibiotics and other things to permeate. So we see that as a side benefit of using this phage in people, and we've actually used this phage in people for personalized medicine. A little bit more on this. It's a little technical, but hang with me. The blue bars are, if you measure for some strain of these bacteria their ability to make piocyanin, in the presence of these various antibiotics, in the case of this analysis, these antibiotics are not killing the bacteria. And in some circumstances, it actually causes the bacteria to make more of the virulence factor. It's not what you would want if you're delivering, say, Cipro to your patient and this infecting strain is in there. On the right, let me go all the way over to this smallest bar on the right. This is the outcome of, if you use this phage instead, it's not only killing the bacteria, but the ones that are left over are very debilitated for piocyanin production. And it works well in the presence of erythromycin which has been used in patients to calm cellular inflammation without actually killing the bacteria. So it's working fine with it. And you'll see that it is reversing that adverse effect of Cipro. So that's very promising. So keep that one in mind. I'll get back to it later. Now I'll go quickly through an example of a phage that interacts with bacteria and selects against antibiotic resistance. I gave you, well, first of all, this is a cartoon from a review paper that I co-wrote with one of my postdocs, Alita Burmeister. The first two examples, these columns, I actually gave them to you earlier. If you find a phage that interacts with efflux pumps, or if it interacts with this capsule, then you could kill the bacteria and shift things to the lower portion of the screen where suddenly they're more vulnerable to antibiotics. They become sensitive. Here on the right is one last example that's going to help me in the next series of slides. There are these components of the bacteria that are quite prevalent often called LPS or lipopolysaccharide. And a lot of phages use lipopolysaccharide as either a receptor or a co-receptor. So if you have a phage specific for that, it'll cause the bacteria to avoid phage attack through mutation that trims down the height of the LPS, okay, to kind of get rid of the portion of it that the phages are using. But that makes them easier to have antibiotic entry if they have shallow LPS. So it's yet one more way to select for antibiotic-sensitive bacteria. So I'm now going to walk you through a couple of, well, about 4 slides from a paper that we published using E. coli as a model system to examine the interaction of phages with efflux pumps. So it goes like this. This phage U136B, we found it on an agricultural farm, and it selects against antibiotic resistance in E. coli. Here on the left is a very beautiful structure of the E. coli efflux pump called Toll-C. So all I want you to know is this upper portion of it is what is exposed to the outer world, and it has a protein called Toll-C. And we know that this phage interacts with that protein for binding. We know that because if you take various what are called knockouts of these bacteria where one gene is missing at a time, you find that the only thing that this phage cares about for this knockout analysis I'm showing you is when you remove Toll-C and you get growth of the phage that is below the limit of detection. Otherwise, it just doesn't care about these other genes being knocked out. The consequence is on the right. So if you grow the wild-type bacteria, E. coli, an engineered Toll-C knockout, or a spontaneous phage-resistant mutant that escapes attack by this phage, they grow fine in the absence of antibiotic and pretty similar to one another. As soon as you throw them into a high enough level of tetracycline, the knockout doesn't grow as expected because it's not pumping the tetracycline out of the cell, but we're also seeing that the phage-resistant mutant can't grow well either. So that gives us confidence that this phage selects, in this example, against tetracycline resistance and shifts it to sensitivity for the remaining bacteria. So the plot thickens for this one phage because it's interacting with more than one thing on the cell surface, and that other thing is LPS that I described earlier. Here's a diagram showing you a side view of LPS where this is the cell surface, and this is what's extending up. So we've found here in the middle all these genes that are important for contribution of building LPS, that if you knock them out, the phage can't attach and grow. It is using this as a co-receptor. So those are color-coded, and you may notice that these actually have something to say about this middle graph, where you can knock out various of these RFA genes that make LPS, and if you knock out the ones that are color-coded here, the phage can't grow. Here's the consequence or the trade-off on the right. So nothing to do with tetracycline now, but if you shave down LPS enough in E. coli through this phage and its use in selection, then suddenly these phage-resistant mutants, they avoid phage attack, but they're quite vulnerable to a different antibiotic called colistin. So this is a cool phage because it's interacting with the bacteria in more than one way, and it's causing 2 separate trade-offs that would work for biomedicine. I can't control whether any of that happens in a patient. Okay? That's the dirty little secret, is that we want this to work in our favor in the way I've described so far, but I can't control which mutations occur spontaneously in bacterial populations. Here's an interesting analysis that if you just take mutants of these bacteria, figure out how did they solve the phage problem. Was it a change in LPS? Was it a change in Toll-C instead? So if you look at the growth of these randomly obtained mutant bacteria in the presence of tetracycline. Here's the ancestral level of resistance to tetracycline. And for this collection of mutants, it's a perfect prediction that if they change their efflux pump to avoid phage attack, they become more sensitive to tetracycline. If they change the Toll-C to avoid phage attack, they become more sensitive to colistin. Here's an interesting outcome. These are phages that changed Toll-C, and if you measure them in this environment, they change LPS, you measure them here, and they actually grow better in the presence of tetracycline. Not great news. Fortunately, there's like a trade-off within a trade-off here. So this graph on the right is showing that the better they are at resisting colistin, it turns out the worse they are at resisting tetracycline. So again, that can kind of work in our favor if you deployed this in a person in a similar way and under a similar analogy. So if you believed everything that I've said so far, this is all in vitro evidence. It's coming from the laboratory. And this is the kind of evidence that we amassed when we started to talk to physicians at our hospital. And they immediately talked to us of the antibiotic resistance crisis and the current patients that they were treating who were essentially currently dying of antibiotic-resistant bacterial infection. And we got clued in that, oh my, this kind of technology can actually help people immediately if they were willing to undergo emergency experimental therapy. So what I'm going to show you now is plenty of evidence for that. For those who are out of options, of course, they would be quite willing to have this experimental therapy because they literally have no other options. And the first case was pretty well publicized. It's been published on now, and I'll go through it quickly. But this was a man who, I think he was in his early 80s, when it was realized by his surgeon that a relatively common surgery, it's not trivial, but it happens quite frequently. It's called an aortic arch replacement, where it's an example of we can, through surgery, help people's lives, give them more functional limbs through prosthetic, swapping things into the human body that are artificial. The problem is they are great substrates for bacteria to come in and cause chronic infections. And if those are increasingly antibiotic resistant, then again, that's the collision course to a problem. In his case, he had a raging Pseudomonas aeruginosa multi-drug resistant infection that was not really detected in its severity until they opened him up and found that it had eroded away part of his sternum. So it was quite remarkable that he was even alive, and he was way too old and infirmed to undergo the surgery again. And we went to his surgeon and talked about this and said, would he be willing? We could try to help him because we have a phage that kills those bacteria, and we've seen in our laboratory that it will synergize with antibiotics to make them useful again in killing the bacteria, whereas they're currently useless. So I wouldn't talk about it had it not worked. So we had published on that, and it got publicity, and it's quite rewarding that we're in his office thanking him for his bravery to do this and giving him a phage plush toy as a thank you. But I've had many letters from his family saying it's amazing the quality of life that he experienced. He eventually did die of heart failure, but he went back to work, and he essentially was unable to work because of what was going on. Now here on the right, I like to highlight the Scientific American article, not because they mentioned us and other work that is going on in this area, but it's a little bit of a dicey title here. So phage therapy actually never went away in places like Russia, Poland, Eastern Bloc nations. They bought into that technology before antibiotics were discovered, and they kept it, and they've still done it. So it's more about, is phage therapy here to stay in the Western world, in Western medicine? Are we turning a corner where we see this as a legit alternative to antibiotics? So let's look at phage therapy treatment that was given to Paige Rogers, who I said her case earlier. I'll give you some details of her now. So before phage therapy, when she heard about her work and her physician reached out to us, literally because of that man's, the social media presence and other things that were talked about for his case, they contacted us about her. So let me go first to here on the left. She had very poor lung function, and you can see over time, I'll describe it in a moment, she was undergoing pulmonary decline. But there's a way of measuring this easily in the doctor's office. It's how much air can you force out of your lungs in the first second. It's abbreviated as FEV1. For people who are prone to have lung problems, like cystic fibrosis patients, it's just a go-to measurement to see how well their lungs are functioning. So she had very poor lung function, poor exercise performance, and her sputum, her mucus in her lungs was just loaded up with bacteria that were causing this infection. So it was rated as +4. But worse off is this. She had no drugs that they could administer to her that were expected to work because the bacteria in her lungs were resistant to all of them. It's called a pan-drug resistant infection. The consequence of that is essentially she was headed towards, at best, a lung transplant. And if that didn't work, then it could be game over for her. So that was happening for very many years as you see this decline in lung function through these FEV1 measures. And we administered therapy to her by putting phage in her nebulizer. So this is the way that she takes medicine inhalation into her lungs, and we just simply added it to it to see whether this would help her. And you could see at the time of the administered therapy, her lung function did jump up, and the measurements became tighter, and it showed us that she was improving in her lung function in response to the therapy. But the better news, even, is that her lung function improves, exercise performance goes up. The bacteria were not removed entirely, but we didn't really expect that to happen anyway. She was so loaded up with these bacteria that we assumed that if we deploy the phage, it'll work to kill most of them, but it'll shift the remaining bacteria to become antibiotic sensitive, and that worked. So most of these antibiotics she became sensitive to for her infection rather than resistant. And if she has what's called an exacerbation, sort of an outgrowth of these bacteria that cause her harm, now she's got lots of options. She and her physician have a ton of options to deploy these antibiotics, and it should help her. So that's a very good outcome. So I don't have time to go into all these cases. And over 20 of them, we've deployed phages, mostly pulmonary problems, so lung infections, no safety problems that we've encountered. I'm only listing the first 13 or so of the 20 here. Many of them have a similar kind of proximate problem with their lungs being very vulnerable to these chronic infections, either because they're a cystic fibrosis patient or they have developed poor lung function over time through what's called bronchiectasis or chronic obstructive pulmonary disease, COPD. So in all those cases, despite their age, I'm listing their gender, mostly that's been Pseudomonas aeruginosa, but occasionally other bacterial targets that we go after that are multi-drug resistant. But it's very satisfying and rewarding to have this work, and we're trying to publish it in what's called a case series, so we can describe all of them collectively this fall. Some evidence from these patients, a reminder here on the left, when they're undergoing therapy, it's usually in a hospital setting. If they're doing fine, we actually send them home, and they can undergo nebulizer therapy at home because they're used to using a nebulizer all the time. But what we can do is track over time if they have these sputum samples that they provide. We can take them in the laboratory and see what are the bacteria that are there, what are the genetic and what are called phenotypic traits of these bacteria. Are they not only dying, but are they changing in a way to make it evident that they've actually experienced this trade-off that the phages have selected for? So some of the evidence from these patients, did their lung function improve after therapy here in red? Yes, on average. Did the load of bacteria evident in their sputum go down? Yes, significantly on average. Lower right, it's basically, for those who can't see it, it's a graph showing that in the case of the people who had multi-drug or pan-drug resistant infections, if you measure that trait for the bacteria that are left over, we often see that they've switched to sensitivity, similar to Phage Rogers' case. And here I have to go to the next slide to describe what is an inflammatory molecule reduction and why is that such a big, significant outcome. That is, if we deploy one of these phages that goes after a virulence factor, there's a way to examine the outcome of that in the laboratory. You can take those bacteria over time from the same patient, and that's what this graph is showing. So you can measure how much piocyanin is being produced by the bacteria in, say, a patient's lung longitudinally. And you're expecting at the beginning that they make a lot of this nasty virulence factor. These are several clones taken at time point 1, later time points, and by the time you march down here towards the end of the therapy, they're not showing any evidence for piocyanin production that is any different than just growth medium for the amount that's in there. So that's a very significant result, and it's also colorimetric, because I showed you earlier that the piocyanin is a bright green color. So longitudinally, that's what we expect. This work is done largely with our physician collaborators at Yale New Haven Hospital, John and Gail. So, that was my bad, sorry. So the other thing that we could do is, it's a little technical, but we can take those bacteria, grow them in the laboratory, spin them down, discard the cells, and that supernatant, the leftover liquid, you can then take that and put that on cells of human origin. And you measure how stressed out are these cells. Are they making any signs of inflammatory response in the presence of this goop that came from what the bacteria produced? And the answer is that decreases through time as well. If you look at certain markers of inflammation, IL-8 and IL-6, after phage therapy for the people who receive this phage, that goes very much away. And that confirms that the cells in the lungs are better off because the bacteria that may be remaining are not antagonizing with those cells in an inflammatory way. What we're doing is a black box, and I fully admit it, right? We're taking laboratory prediction, expecting it to work in people, and the human body is a very complex thing. So fortunately, so far it's working. Can laboratory data predict patient outcomes? Well, I showed you that they could, but we really have to know how often that happens. So now I'll segue a little bit to the problem that we're facing globally, the SARS-CoV-2 pandemic. Some of my work that I'm not talking about today, I have a long-standing interest in the emergence problem, long before the current pandemic occurred. So we've done lots of laboratory studies, for example, to if you think about how did HIV come into the human population? It was through host shifts, where other primates, chimpanzees, Sudi-Mangabe monkeys, several times independently, the viruses that were in them shifted into humans. So why is that? So there's some undercurrent of, if a virus is very good at using multiple hosts, it could be better prone at spilling over into another host successfully, like humans. So we've tested a lot of that in the laboratory, mostly in a theoretical and conceptual way. More recently, we're digging into the emergence problem a little more dedicatedly by looking at the pathogen itself. So we do work on arthropod-borne viruses, especially dengue virus, that this projection map shows the warming world and how in the future, there are a lot of places that are going to be more vulnerable to mosquito-borne infections because the mosquitoes can survive overnight at a higher temperature. Okay, it's not good news for the East Coast of North America, for example. So we would like to better predict how does dengue virus, for example, emerge in new places, and how much does climate change contribute to that? And here on the right is the thing that we're most doing in earnest in response to the pandemic is the concern of zoos and aquaria that their animals actually might get this virus from people, or that if it gets in the animals, it spills back into people, which is a legitimate concern. And in our case, we're looking at it in whales and dolphins, the vulnerability of them to SARS-CoV-2 in collaboration with Mystic Aquarium in Connecticut. I tell you all that because to me, those questions of importance are not that different from what I've been talking about all along, but in a slightly flip it on its head way. So our goal is to better predict phage potential to broadly kill target bacteria. I told you that that's what I want to achieve. But you have to think about, can you push that to the extreme and get a bad outcome? If you've got very generalized killing of some target bacterium, are you setting the stage for a host shift of those phages to move into what are called commensal bacteria that actually do you some good, right? And that's not what you would want to happen. So that's, I think, an important question, is will phage therapy cause issues with the microbiome. Quick reminder, that's certainly what broad-spectrum antibiotics do anyway, right? You deploy them and they kill the target bacterium, and meanwhile, they get rid of a lot of your species in your microbiome that are doing you good for human health. Fortunately, you can mostly recolonize those, and I won't get into that tonight. So it's not as if this is an in-game problem, game over. It's more about we need to be informed as we move forward with phage biotechnology. And as we're doing this, the full disclosure is that I helped found a startup company that has a mission to try and get phage therapy to work in a commercial way. And to do that, you can look at different pathogen targets. Felix Biotechnology, named after Felix Durrell. So that's where that comes from. So we can discover phages and characterize them. We can use them in personalized medicine. That's preclinical. So we're doing a lot of that in P. aeruginosa, for example, but also in MRSA cases. I didn't talk about that tonight. But the idea is to try and shift them into this column, where you do a clinical trial, which is something that would say in healthy volunteers, could this even help them be protected against future bacterial infections if you give them a phage that's safe to enter the human lungs and it pushes out the possibility of more bacterial infections coming in? So that's what we're doing now at our hospital. It's abbreviated as SciFi. It's the Cystic Fibrosis Bacteriophage Study at Yale. This is literally healthy volunteers who have cystic fibrosis, and they're quite vulnerable to these infections, but we want to track them over a multi-month period that if they see the phages that we use to counter these bacteria, will they be prophylactically protected better than others in the trial who are receiving a placebo? Okay, and it's called I think the word is crossover. I can't remember the exact term. So nobody has really denied the therapy. They just don't know when they're on a placebo versus when they're actually getting the phages. We have, from Yale, the resources to start a center that hasn't gone live yet, but we have started it. And the idea is to try and build more of this out. Continue doing the personalized medicine, continue doing clinical trials, come up with a less menacing symbol for the center. So if you have any ideas, I think that snake doesn't really look that inviting, but the point is we're doing that, and we're trying to take this into countries and places where already the high cost of drugs is very prohibitive for them to be purchased and used. So in many parts of the world, including some countries in West Africa, for example, we've gone there and held workshops for physicians and researchers to start learning this technology early, seeing how it can be done safely and effectively, and that has been working very well. Last thing I'd like to say is that, unfortunately, this is not just a human problem. So the antibiotic resistance crisis crosses over into bacteria that are multi-drug resistant, that are killing animal and plant agriculture and killing in aquaculture. So that threatens the food supply. And you don't want this to be occurring, obviously, and we would like to see whether this would be a good alternative. I mentioned already Mystic Aquarium and our work in collaboration with them for virus emergence. But in other settings, in zoos and aquaria, they're also worried about multi-drug resistant bacterial infections because they're already seeing it. So folks in my group, I wasn't involved, but they worked with veterinarians at Moat Aquarium in Sarasota, Florida, because one of the star attractions of that aquarium was this huge sea turtle that had a multi-drug resistant infection caused by a bacterium that's ordinarily very benign called Citrobacter frundii. We knew nothing about that bacterium, but we were good at phage hunting, finding the phages that would help it, and it saved this animal's life. And of course, they were quite appreciative of that. Now, the last thing I'll say about that is why would you spend a lot of time and attention trying to save a sea turtle? It's because you can't take them from the wild very much anymore. A lot of the animals that are in zoos and aquaria, you literally cannot take them from the wild anymore. So they would spend a lot of money to try and keep these animals alive because it helps the mission of the zoo and aquarium sell tickets to keep them alive. Okay, so I've said a lot, I've come to the end, and I'd like to acknowledge the people who actually did the work. So my lab group through time has just been populated by fantastic people. I wish I could name all of them, but it would be way too long of a list. Here on the right, I'm showing most of the folks I highlighted today. You can see them by name, who I mentioned before. Barbara Kazmierczyk is one of my colleagues at Yale Medical School, who has been one of the very many people, George O'Toole as well, who have helped us by providing, say, strains of bacteria that we can study in the laboratory to further this technology. Rob McBride and Natalie Ma are formerly at Yale, and now they're at our company in San Francisco, Felix Biotechnology, as the CEO and a project director. So the last thing I'd like to do is acknowledge Deepak Narayan. He was the surgeon who helped us on that first patient case. And believe me, when we sat down and talked with him about this, he must have perceived this was a very wacky idea. Right? People were just not doing this yet. And he believed that his patient was desperate enough that something else had to happen. And he wasn't quite sure what it would be. And he figured, okay, what we are pitching to him should be safe enough to try it as long as we watch the patient like a hawk to his response. And it worked. But unfortunately, Deepak died of cancer in 2018. And I am ever grateful for him for opening the door for the other work that we've done because he believed in what we could do for that first case. That's it. Thanks for your attention. Hopefully I left a little bit of time for questions.
Yeah, we do have a few minutes for questions from here in the auditorium, or they can come in online as well. So if you have a question here, please just raise your hand.
As they say, there are no stupid questions.
Yeah.
Just stupid answers for me, which is entirely possible.
Hey, thank you for such an insightful and thought-provoking talk.
Thank you.
I know for HIV and other viruses that combination therapy, so drugs that attack different parts of the life cycle, have been really instrumental to combat the problem of resistance and evolutionary divergence. And I wonder if you think about having multiple phages targeting the same bacteria to combat the same problem of the bacteria becoming resistant to the phage.
Absolutely. That's actually a more popular idea than a lot of what I told you tonight. Right? That's called a cocktail. And I think there's just a keen interest or an assumption actually that it would work. So the interesting thing about cocktails is that they generally do debilitate a target bacterial population. They kill it better than one phage alone for the reasons that you said. What we care about as evolutionary biologists is what happens next. So often those bacteria may change genetically in a way that makes them more difficult for future phage therapy or any other therapy to kill them. They sort of hunker down in a way and evolve traits that are more resistant to all therapies. So that worries me. I can't predict those genetic outcomes better than I can when I'm using one phage or a series of phages in sequence. So largely what we've done more recently is go to the single phage if we could use it, or deploy one phage, look for the response in the patient to be improved in the way we predicted, and then move to another phage if we say want to push down at that point on antibiotic resistance and switch it to sensitivity. So that is easy to say. It's very hard to prove which strategy is better, and I'm not going to claim that there's one universal strategy that works. I think it's very dependent on what's the target bacterium, what part of the body are you trying to go after, et cetera, et cetera. So I think what you're going to see from my lab and others are giant studies about lots of ways to deploy phages and looking at the full genome sequences of the bacteria that evolve resistance and trying to grapple with this problem about, what for this one system that we're trying to work on is a better strategy relative to another. Hope that, I'll try to be more brief with my answers. How's that? Somebody, yeah. Hey.
Paul, great talk.
Thanks.
So my question is about delivery of phages in phage therapy, which is like obviously one of the biggest barriers. So you talked about the person with the aorta device.
Yeah.
So I guess one, how did you deliver phages to the aorta?
Yeah, that's interesting.
I guess.
Want me to start with that?
Yeah.
So the idea was we knew the site of the infection through various studies that they'd done, imaging, right? So they were going to take a syringe and put it through the chest wall and deploy the phage as close as possible to the site of the infection. Literally, they were in the room doing the surgery, and they encountered too much scar tissue, and they couldn't reach it. So they took advantage of a quite gruesome feature of this man's infection. He had a fistula, an oozing wound, because the infection was so bad it had broken through his chest wall and it was oozing out. And they were able to take, I think it was, the story goes like this. It's in the paper, actually. Sterile gauze soaked in the phage antibiotic solution, placed on his chest, and kind of through a combination of trying to get it into the fistula opening and just gravity, pulling it down to, it worked. It worked with one administered dose, actually, in his case. What was your second question?
So I guess my second question, so that's like an invasive method of
Yes.
delivering phages as opposed to like a nebulizer. Do you worry about the host immune response potentially, like if you had to re-administer the same phage, if the infection reemerged,
Yeah.
that the immune response could come and be the next hindrance to phage therapy?
Yeah, I worry and I don't. So I think what we have seen is that if you knock a bacterial infection down enough with phage, and it's synergizing even more with antibiotics, but you're not getting rid of it, that's actually when the immune system can work against those bacteria. And you get a better outcome because you've calmed the infection down enough for the immune system to actually handle it. But your example is, can I go in with the same phage more than once? And there I have only anecdotal evidence that yes, we've administered the same phage to some people like 3 months after they got the first series of doses. And we warned the physician that it wouldn't work for the reason that you said. Like, nah, it's never going to work. Okay, you got your approval from your hospital, sure. And then it works, and we're as mystified as anybody why it works. So I think the punchline is that it depends on the phage and the bacterium, about how robust of an immune response do you get. And then many other things happen. We're working with Jen Bomberger's group at Pittsburgh about some phages, maybe many of them, enter human cells, but can't replicate. And they'll enter human cells to the point of causing those cells to have an interferon response, which is used against generally killing viruses of humans and bacteria. So it starts to kind of turn on immune responses in a way that's working to our benefit synergistically when you deploy phages. I don't have the time to go into it too much, but I think there's been many historical instances of people head scratching about why did phage therapy work so well? And now people are digging into the exact mechanistic reasons for it. So your question is spot on, and I don't have a great answer for it. Maybe that's why I'm dancing around it. But it's part of ongoing research about how much can the immune system catch up.
Hi, thanks for sharing all that with us. My question is actually one step upstream of Danielle's, which is how costly and labor intensive is it to create these phage cocktails that are pure enough and dense enough? Also, how do you know how dense they need to be to administer to patients?
Good. So growing phage in the laboratory is super easy, as many of the people here in the audience know. You can grow a huge batch of phage very cheaply, very inexpensively, because they just make copies of themselves. But what you have to be careful with in that phage solution, right, it's called a lysate, is if you want to then put it in the human body, it's also got a whole bunch of cell debris because they've killed these cells. So you have to put it through a dialysis process to remove that. And if you don't do it carefully enough, you're going to end up delivering to the person what are called endotoxins, because these are that's what's going to happen in response to this flooding into the human body of a bunch of bacterial cell debris, is it's going to cause you to go into shock, because that's your response to these endotoxins. That was certainly our worry with the first few patients that we administered therapy to. In my opinion, I think it's an enduring worry if you just put phages into the bloodstream. That is a very popular way that some people want to develop phage therapy. I'd rather get closer to the site. So the process is quite reliable to do it safely. And in our case, with the first few instances of this personalized medicine, we sent our preparation to 2 laboratories that tested it themselves and reported directly to the FDA to confirm what we had measured and claimed was a pure lysate that didn't have all these endotoxins in it. So my point in saying all that is there are some no therapy is without some risks. And people have to take sometimes better attention when you have a super cheap therapy. You have to be even more careful because kind of anybody could do it. And that's why we would like to kind of create more working knowledge and safe. And a lot of people have the same goal. So I'm not saying we're the only ones doing it. There are very many laboratories that are trying to share good protocols on doing this. Does that help with your? Yeah. There you are.
Hi.
Yes.
Thanks so much for your talk. That was great. You mentioned that self-amplification is one of the things that makes this therapy attractive.
Yeah.
But we also are sure what are some of the conditions that will cause maybe select for phages that can go into these commensal microbiota.
Yeah.
And host shifts in general are of great interest. So what are some of those things that what are some conditions that you think will cause these host shifts? And like, do you think that there's going to be these common mechanisms that inform that between systems?
Yeah, so I guess the expectation is that the closer some new host is to the old host for a virus shift to occur, it makes it more likely. In many cases, I think I would buy that argument. So if you are going to deploy phage therapy in a portion of the body going after a pathogen that is a close relative of a commensal bacterium, that's where I think you have this higher likelihood. But I think you know that. I'm sure you know that. We, so far, have had the luxury of mostly working in a part of the human body that's not very impressive for its commensal microbiome that is expected to do people good, at least as far as I know. It would be vastly different when we start going into the gastrointestinal system. So I think that the interest in all of this and the very many strategies that people are kind of trying to come up with, that shouldn't be ultimately a barrier. I mean, those bacteria are attacked by phages all the time in your body. And I don't know that necessarily some new invader that I put in that's going to limp along in trying to use those bacteria. Why would I expect it to even outcompete the phages that are already in your virome that are infecting those bacteria? I think that's often what people don't talk about. And maybe they don't talk about it because those experiments are hard to do and people haven't really kind of done much of that. Does that kind of make sense? Okay. I saw somebody.
Unfortunately, we're out of time. Those of you who are lucky enough to be in the auditorium obviously can hang out for a little while.
Okay.
But let's please thank Dr. Turner. Maybe we can get some of the questions online answered
Sure.
later. So,
Thank you for your attention.